Hi everyone, and welcome to our second lab. This one focuses on the monohybrid and dihybrid crosses. Now keep in mind a lot of what we covered today you will have already gone over in lecture, but honestly that's the whole point of labs, is to help solidify your understanding of what you cover in lecture and kind of help supplement it so that you have a better working knowledge of the concepts that you're encountering in genetics. Okay? Now, when we talk about genetics, ultimately we are looking at the study of heredity and variation. So that means what are you or the organisms we look at inheriting in terms of their genes and the variety of different, types of genes and phenotypes that you can encounter. Now, when we talk about genetics, you always hear the name Gregor Mendel as the father of genetics. And I always like to ask the students, how did Mendel study inheritance? Why is he the father of genetics? What did he focus on? Now, Mendel was a monk who studied pea plants, and he looked at the fact that all of these different pea plants, they showed a lot of variety. So he would look at and study the different flower colors, the colors of the peas themselves, even the texture where, some plants had wrinkly looking pea pods and others had, nice smooth peas, and things like plant height and all of that kind of variety. Now, instead of studying plants, what we do, we're studying flowers and peas. What we do in the genetics lab is we're going to use fruit flies in order to demonstrate some of the concepts. Now, ultimately, when you study these organisms and the variety of things like the pea plant colors and textures, or the fruit flies, their eye colors and wing types, you end up seeing how the traits are passed on to their offspring from the parents and how predictable it actually can be. Now, ultimately, this inheritance is guided by genes, and genes are the unit of inheritance that are found in pairs. And I want you to highlight the fact that all of the genes in an organism you're going to find in pairs. The reason that is because you get one from mom and one from dad. So that's why you have, for instance, in the figures over here, you have homologous chromosomes, meaning for each gene you have two different pairs because one is inherited from the male parent and the other from the female parent. Now, ultimately, these pairs of genes and pairs of chromosomes, they get separated when that organism then goes to make its gametes. And we mentioned what gametes are in the first lesson, so make sure you write down the definition of gametes, which are sex cells, okay, not sex cells as in, what they always say about the media and advertisements, but gametes are your sex cells, meaning the sperm or egg that is produced to then, pass on genes and traits to the offspring. Now, keep in mind, we also mentioned when we talked about genes that there are alternate forms of genes, and what that is called is the allele, okay? So you have more than one form of gene. So for instance, white eyes versus red eyes in the fruit flies, that would be considered two different alleles, A-L-L-E-L-E-S, okay? So the alleles are simply the alternate forms of genes, and then one allele can be tall versus the other allele for a gene being short. Red eyes versus sepia, white eyes versus red eyes, all of those are examples of different alleles for genes. Now, when we talk about alleles, keep in mind that for each trait, there's basically more than one form of the genes coding for that trait. So like we were just saying, tall versus short. In flowers, you'll see them say red versus white or purple versus white flowers. Those are alleles. Now, when you inherit your genes from your parents, you end up inheriting one copy of that gene from mom and one copy from dad for each trait. So when you're looking at a particular trait, you're going to have two alleles, okay? So any genotype for a trait, such as when we talk about the fruit flies, we'll look at the trait of eye color, and you'll have two alleles for that fly, one from mom, one from dad. So for instance, they may get a red wild type allele from mom and a sepia mutant allele from dad. So ultimately they have two alleles. Now, when you're looking at the inheritance of those alleles, that offspring can either end up being homozygous, heterozygous, okay? Homozygous or heterozygous. Sometimes we call heterozygous hybrid. Now, if a genotype is homozygous, homo means the same, so that means that both alleles for that trait will be the same. Okay, so both alleles for the trait will be the same. For instance, if we're looking at a sepia fly that has the dark eyes, it's sepia with dark eyes because both alleles are the sepia. Okay, so for instance, you would see it written out as SS. Now, when we talk about fruit fly alleles, what we end up doing is for wild type alleles, we show plus marks. So homozygous for the fruit fly with eye color would either be SS for sepia or plus plus, okay, 2 plus marks for homozygous wild type. Now, if you're looking at sepia versus wild type and a fly has a heterozygous genotype, it would mean one allele is wild type, the other allele is sepia, because heterozygous means that for that trait, there are two different alleles, okay? Again, that could be one wild type, one sepia, or if you're looking at height, like the height of the plants with Mendel experiments, it would be one allele is tall, one allele is short. Again, the reason why you have two is because one is from mom, one is from dad. Okay? Now, when we talk about alleles, you also have to know the terms dominant versus recessive. And dominant simply means that anytime that allele is present, that is what you will end up seeing in the phenotype. It masks or suppresses the recessive partner, whereas you will only see a recessive color or phenotype, physical appearance, when there are two recessive alleles. The moment you get a dominant one, you're seeing dominant. So for instance, if we go back to what we drew out already, the homozygous and the heterozygous, wild type in the flies is dominant. So we put wild type in flies is dominant, whereas the mutants are recessive alleles. So if we look at the genotypes we have on this slide, this one, what would it look like? Well, do you see any dominant plus marks? No. So that would look sepia. Whereas this one and this one both have at least one plus mark. So both of these would look wild type. And what's the wild type eye color? Red eyes. Okay? So make sure you're very comfortable with that terminology because it will keep coming back up. Now, when we talk about the inheritance of these different genes and alleles, all of those genes and alleles for an organism are ultimately found on its chromosomes, okay? Now, each species has a set number of chromosomes that defines that species, okay? For instance, humans are defined by the fact that they have 46 chromosomes. Now, again, you find those chromosomes in pairs, so you'll always either hear it as humans have 46 chromosomes or humans have 23 chromosome pairs, okay? Now, when we talk about chromosomes, you'll either hear a diploid number or you'll hear a haploid number. And the difference between the two, when you look at diploid number, the way we write that out, that question mark, you replace with 2n, whereas the haploid number is represented by n, okay? So the diploid number is maintained by mitosis, okay, mitosis, whereas the haploid, which is the half number, is maintained by meiosis. Okay, so you see here, meiosis here, mitosis here. Okay, so the human diploid number would be 46 chromosomes, and the human haploid number, meaning the half number which you find in the gametes or the sex cells, would be 23. Now, it's important to kind of figure out why we end up having that, diploid maintained by mitosis, haploid maintained by meiosis. And if you think about it, mitosis is when your somatic, meaning your non-sex cells, so all the other cells in your body that are not your sperm or egg cells, those all replicate or divide by mitosis. So for instance, your skin cells. The reason why they need mitosis is you always want all of your regular body cells to have 46 chromosomes in them, the diploid number, 2n. Otherwise, you're not human if you don't have the proper number of chromosomes. Okay, now there are exceptions to that. There are unfortunately disorders and different diseases that will lead to a slightly different chromosome number, and they are still human, okay? But in the scientific definition of chromosome number, the reason is to maintain proper human functions, and each species has its own number, okay? So once that species deviates too far from its usual number, then you end up with a different species, okay? So now with mitosis, those somatic cells, or all the body cells, will double their DNA, then split in half, okay? And they'll end up with the proper 46 chromosomes. Whereas the haploid number is maintained by meiosis, which is the formation of sex cells, okay, formation of gametes. And the reason why the haploid number has to be maintained by that form of mitosis is that ultimately you want the sperm cell or the egg cell to have only half the number of genes, because what's that sperm cell or egg cell going to do? It's going to end up going through fertilization, and it will fuse with another sex cell. So that's why it needs half the amount of genes, so that when they fuse together, what do they have? The total diploid number again. So a sperm cell will have 23 chromosomes, the haploid number, and the egg cell that the sperm cell will fuse with will also have 23. When they fuse together, they now make an organism with the proper 46 chromosomes, okay? Which we refer to as also 23 chromosome pairs, okay? Again, they got 23 chromosomes from mom and 23 individual chromosomes from dad to make the 46. Now you'll notice that I wrote a question on this slide, so kind of to reinforce the idea of haploid gametes versus the diploid number. If you have an organism that has a gamete with 30 chromosomes, then how many chromosomes will be in the somatic cells of the body? So please make sure to write down that the gamete is the sex cell, either sperm or egg, and the somatic cell is the body cells. So for instance, your skin cells. So if the gamete has 30 chromosomes, for instance, the sperm cell has 30 chromosomes, that means that during fertilization, it will fuse with an egg that has 30 chromosomes. So that somatic, the somatic cells of that organism will be 60 chromosomes. Okay, so the haploid number is 30 and the diploid number is 60. Okay, twice that amount. If you have any questions or are uncertain of anything we covered in this cell, please make sure to contact me in the Remind app and I will go over it again in other words, or I'll give you another practice problem. Now, when we do any crosses with the fruit flies, you'll hear us say certain terms related to the generations. You have the P generation, the F1, and the F2. So P stands for parental, meaning these are the initial homozygous parents. Which we'll go over with regard to the fruit flies in the next slide. Those parents then have their offspring, which are the F1 generation, so F standing for filial, which means the children, the offspring. And because the parents were homozygous, okay, so let's say homozygous capital letters, homozygous little letters. And again, I'm not using the normal, W or whatnot. We'll just go based on this. So parents, both homozygous, and then all of the filial one or first generation of offspring will now be heterozygous or what we call hybrids. One allele from mom, one allele from dad. And then you end up with the F2 generation standing for filial 2, which is the next set of offspring. And again, we're going to go over why you see 3/4 and 1/4 in a little bit. But for now, just this slide is just to show you that P stands for parental. Then you have F1 and F2 generations. Now, the first cross that we would normally do with the fruit flies is the monohybrid cross. And whenever you see a word in biology, break it down so that you can figure out what it means. So when you see mono, that means one. So you're only looking at a single trait of the fly. And then hybrid, we've already mentioned, is heterozygous, meaning you're looking at a trait that can have a dominant and a recessive allele. And you're crossing the hybrid flies together, the heterozygous flies together. Now, in the classroom, when you want to cross these two heterozygous flies together, you first have to make them. So how would you make heterozygous flies? Well, there's no Tinder for fruit flies. So what you have to do is you start with homozygous flies from the manufacturer. So what you do is you have one parent that's homozygous wild type and you cross it with one parent that's homozygous recessive for the trait that you're looking at. So in the example on this slide, the trait that we would be looking at in this monohybrid cross is wing type. So you would cross in your first parental set of flies the homozygous wild type flies that have nice long normal looking wings with the fully recessive little apterous fly which has no wings. And again, apterous, the way you would write that out, apterous fly, meaning no wings. Okay, so you would cross those two flies together. Now notice in this picture you have two flies. I want you to stop everything, hit pause, and send me a Remind app message telling me which of the two flies on this slide, the wild type or the apterous, which of these two is male and which is female, and how do you know that? Okay, so tell me what in the image demonstrates male versus female. What is very obvious in this image to kind of help you distinguish? Okay, so make sure to send me that reply in the Remind app right now as soon as you get to this slide. Okay? Now, once those two flies, the parental flies, and I'm going to say two, but you would actually have multiple of the wild type mixed with multiple of the Apterus. Now, once those parental flies make their offspring, aka, meaning they have sex with each other and produce new flies, well, those new flies that they produce are now the F1. And because you mixed a homozygous wild type fly with homozygous Apterus, okay, so homozygous wild type, which we show with the plus symbol, with homozygous Apterus, which we use with the mutant letter A, because you mix those two flies together, all of the F1, what they're doing is they're getting one allele from parent number 1, one allele from parent number 2. So that means all of the F1 will be heterozygous or hybrids, okay? That will be the F1, and then you take those F1 flies and you cross the F1 flies together. I know, technically a lot of them are siblings, but flies don't care about that, okay? You cross those together and those will give you the F2 generation. Okay, and you're able to use Punnett squares to help visualize the probability in each of these generations. So now to make this a little clearer, we're going to draw out the Punnett squares right now. Okay, so like I said, the first thing that you're doing is you're starting off with the parental generation. And in the monohybrid cross that we were just looking at, okay, what did you have? You had homozygous wild type fly crossed with homozygous recessive. Okay, so you can remind yourself that, that's one parent crossed with the other. This is the wild type parent crossed with the Apterus parent. Now, when that parent goes to make its gametes, meaning its sex cells, the first parent, what can it give as a gamete? It only has one type of allele. It's homozygous wild type. So it has to give the offspring the plus, right? The wild type. What about the second fly? It only has Apterus alleles. So it has to give that offspring an Apterus allele. So parent number 1 gives a wild type allele. Parent number 2 gives an Apterus allele. So that's the genotype for wings. Again, remind yourself we're dealing with wings here. So this fly offspring, what do we call it? That is now the F1 generation of flies. Okay, all of the F1 have the genotype of plus A. So that's the genotype of the F1 is plus A. And the phenotype, what's the phenotype for all of those? Well, as soon as you see that wild type allele, wild type is dominant. So they all have wild type long, normal looking wings. Now what you want to do to make the F2, you cross two of the F1 flies together. So you cross one heterozygous F1 fly with another heterozygous F1 fly, okay, in order to make the F2 generation. And the way you can predict what the F2 generation is you make a Punnett square, okay? And what you do is you have one parent's gametes up top here, and then the other parent's gametes on the side here, okay? So you can remind yourself parent 1 and parent 2. Now with the law of segregation, remember, when a parent goes to make gametes from its genotype, those alleles separate for that trait, right? So what alleles can it give to a gamete? Well, the first gamete can be wild type, the second gamete can be apterous. Okay, so that's parent 1's possible gametes. This one over here will be parent 2's possible gametes. Now, what can our parent 2 give? Well, it can either give a plus symbol or an A, right? So a plus symbol or an A. Now all you do is you go square by square, combining parent 1's gametes with parent 2 gametes. So in this square, parent 1 is giving a plus symbol wild type. Parent 2 is giving plus symbol wild type. In the next box, you have parent 1 giving letter A and parent 2 giving the wild type plus. Now we usually write wild type first in the genotype, so I'm going to put plus and A. Okay, notice that each square represents the F2, the new baby, I'll call it a baby, the new offspring's genotype. And for that trait, we're looking at one trait because it's monohybrid. That trait, you have to have 2 alleles, one from mom, one from dad. So now we finish out the bottom. Parent 1 in this square is giving an allele of wild type. Parent 2, an allele for apterous. And the last one, both parents are giving an allele for apterous. Okay? So the F2 generation now, you notice F2, sorry, my pen does not want to keep writing. It's dying over here. Okay, so our F2, you notice that there are 4 offspring in that Punnett square. So the F2 genotypes, we're going to write first. You notice you have the first square had plus mark and plus mark. Both of these middle 2 had plus A, okay? And then the last option was AA, okay? So you had 1 out of the 4 were homozygous wild type. 2 out of the 4, or 1/2, was the hybrid or the heterozygous. And then 1 out of the 4 was fully apterous. Okay, so the genotype ratio was 1 to 2 to 1. Okay, and you could write the genotypes underneath to remind yourself what genotypes you're referring to. Then you also have to keep in mind the F2 phenotypes for the monohybrid cross. Okay, so what does plus plus look like? Well, that looks like wild type wings, right? But what else looks like wild type wings? Plus A, right? The heterozygous, because as soon as you see any plus mark, it's gonna look wild type. So now you have 3 out of those 4 boxes, 3 out of those 4 options look wild type. So all of these guys here will look wild type, whereas the only one that will look apterous because it has fully apterous recessive alleles is the last one. So 1 out of 4 will look apterous. And remind yourself, what does apterous look like? No wings, okay? So 3 out of 4 phenotype-wise will be wild type. 1 out of 4 will be apterous. So the phenotypic ratio is 3 to 1, wild type to apterous, okay? If you have any trouble following the P generation, the F1 or the F2 generation, please let me know. I could ask you to cross the flies. I could ask you about the genotypic ratio. I could ask you about the phenotypic ratio in a monohybrid cross. Okay, if you'd like any examples or practice problems, please contact me in the Remind app. to let me know. So now what you noticed in the last slide when we had the monohybrid cross results of the F2 was that both the homozygous dominant wild type and the heterozygous, that plus A genotype that we showed, both of them, what did they look like? Well, they looked like wild type. So when you encounter a wild type looking fly, a lot of times, you can't be sure, well, what's its genotype? Because the dominant wild might be masking a recessive allele. So if you ever want to figure out the genotype of a dominant looking organism, if you want to figure out what its genotype is to see whether it's homozygous or heterozygous, what you do is a test cross. And a test cross is when you cross that dominant looking organism with a homozygous recessive organism. Okay? So for instance, if we were dealing with the monohybrid that we just talked about, the wild type versus apterous flies, what you would do if you had a wild type fly and you wanted to figure out, is it homozygous wild type or is it heterozygous wild type, you would cross it with an apterous fly, which is the little a recessive, fully homozygous recessive genotype. And then you would look to see whether or not the offspring of that test cross are all the dominant phenotype, or do you see a mixture of dominant and recessive looking phenotypes? Okay, so basically with a test cross, if you see that all of the offspring are the dominant looking phenotype, for instance, we see that in this example here. Tall is dominant, dwarf is recessive. When you cross the unknown, so we can refer to this one, normally it would be the dominant looking one is an unknown genotype. And same here, your dominant looking one is an unknown genotype. What you do is you cross it with the recessive. Okay, so you cross it with the recessive in a test cross, and you see, if you see that all of the offspring, 100% offspring look dominant, then that tells you that your unknown genotype was homozygous dominant. Whereas if you see half dominant, half recessive, then that tells you that the unknown genotype was heterozygous. Okay? So in a test cross, again, you're crossing an unknown dominant looking organism with a fully recessive one. If you see all dominant offspring, that tells you that your unknown genotype was homozygous. If you see half and half in the offspring, then it tells you that you had a heterozygous organism. Now we're going to move on to the dihybrid cross. And again, break down the word. Now instead of mono, you're seeing di, which tells you that you're looking at two traits at the same time. So the way Mendel did this kind, he was looking at seed color and seed texture. For us in the genetics lab class, the way that we would look at this one is to look at two traits of the fly. So we'll look at eye color and wing type at the same time. Now again, you can track this out with a Punnett square, but keep in mind it's going to be a little more complicated. We're going to show it on the next slide. But when you do this, you have to keep in mind that each gamete, meaning each sperm or each egg, must have one allele for each trait. Okay, so the offspring will end up having two alleles per trait because, again, remember, you get one from mom, one from dad. Okay, so you end up with the answer to these questions. Okay, you have 2 alleles per trait. Okay, and then per organism for dihybrid cross, you're going to have 4 alleles. Okay, 2 alleles for trait 1, and then 2 alleles for trait 2. Okay, and a lot of times I like to kind of write it out, trait 1, trait 2, to remind myself what's going on. And for the dihybrid cross, we're going to look again at 2 different traits. We're looking at eye color, so the mutant will be sepia for that, and we're looking at wing type. So we're looking at sepia and apterous flies. Okay, so now again, keep in mind for dihybrid, we're going to be focusing on 2 traits now. So it might look a little scary, but it's actually quite easy if you keep everything nice and organized. Basically, keep reminding yourself that you're looking at 2 traits. Now, because it's a dihybrid, again, you want to be crossing 2 hybrid flies together, but just like with the monohybrid, you have to first make those hybrids, those heterozygous flies. So you start out with your P1, your parental generation, being homozygous stock flies, meaning stock, they're coming from the manufacturer. So you want to start out with homozygous flies, and we cross an apterous fly with a sepia fly. Now keep in mind that flies are named after their mutation, but we're looking at 2 traits. We're not just looking at 1 trait. So when we look at our first fly, when it's called apterous, that tells us that the wings are mutated. But what's our second trait that we're looking at? We're looking at eyes as well, right? Well, do you see any word for eyes in the name apterous fly? No. So what does that tell you about its eyes? Its eyes are wild type. Okay, so when you draw out the genotype for this apterous fly, you would first draw out the homozygous wild type genotype for the eyes, and then you would write the alleles for wings, which in this case for apterous would be 2 lowercase recessive alleles. So again, notice that that genotype has 4 alleles because we're dealing with 2 traits. Each trait, so eyes and wings, each have 2 alleles because that fly got 1 allele from mom, 1 allele from dad for each trait. Okay, so then we cross it with our homozygous sepia fly. Now sepia describes the eyes, right? So mutants are recessive homozygous mutant for the eyes. What about the wings? What would the wings be? Well, do you see any word for wings in the name of that fly? No, it's just sepia for the eyes. So that tells you that the wings are wild type. Okay, so just like we said before, you have your 2 traits, each having 2 alleles. Now, when you go to make the F1 generation from those parent flies, well, what do you notice? Again, think of the traits separately. So for eyes, these offspring can only get a plus or a plus. So no matter what, the F1 will get 1 plus allele from parent number 1. And what eye color allele can it get from the second parent? Either lowercase s or lowercase s. Either way, it's getting an S as the second allele. Okay, then you do wings, the second trait. So what allele can it get from parent number 1? Either an A or an A. So it's getting an A from parent number 1, and from parent number 2, it can only get a plus or a plus. And again, we usually write the plus symbols first ahead of the mutant allele. Okay, so again, keep going based on each trait. So all F1 will be completely heterozygous. So they'll be heterozygous, 2 different alleles for eyes and for wings. Okay? And so you cross the F1 with each other. Again, I know gross, some of them are siblings, but that doesn't matter to flies. Okay? And then you move on to see what kind of F2 this heterozygous cross can produce. And so to do that, we're going to make a big, scary looking Punnett square. Okay, the dihybrid Punnett square looks very scary, I know, but again, it's very doable if you keep it nice and organized. Okay, so we're going to make a big Punnett square here. Okay, and we're going to separate it based on the different gametes that each parent can give to the offspring. Okay, so we've got to just draw this out first. We're going to have parent number 1's gametes over here, and then parent number 2 along this side. Okay. All of the things we're going to write now will be the 4 gametes possible for parent number 1 to make. Okay, so in this top area, we're going to put the gametes or the sex cells that parent number 1 can make. Now, when parent number 1, the apterous fly, the heterozygous fly goes to make its gametes, when you make gametes, sperm or egg, what do you know about your alleles? Well, they separate, right? We did that as the law of segregation last time. And so each gamete that this parent makes must have 1 allele for eyes and 1 allele for wings, right? So we're looking at this parent. What gametes can it make? Well, 1 allele for eyes, 1 allele for wings. So the first gamete is plus plus. Its second gamete is again 1 allele for eyes, 1 allele for wings. So 1 allele for eyes, it could give a wild type. Second, now we go 1 allele for wings. We already have the first gamete plus plus, so this one instead can give little a. Then that fly can also, for eyes, if it doesn't give a wild type allele, what does it give? It gives its recessive allele. Okay? And then for wings, it can either give wild type or A. So we'll give wild type here for wings. And then the last one, eye and wing, it can give its recessive, or for wings, it can also give recessive. Now you'll notice that our F1 parents, parent 1 and parent number 2, so this is parent 2, they have the same genotype, so they can make the same alleles and the same gametes. So parent 2 will also make 1 gamete that's plus plus, 1 gamete that's plus A, 1 gamete that's S plus, and 1 gamete that is SA. Okay? Now again, normally you would put plus in front of, you know, the recessive, but in this case we're going to keep them organized as eyes and wings. Okay, so that you remember gametes are 1 allele for each trait. Now you put the gametes together. So this first gamete fuses with this second gamete, so parent 1's sperm, for instance, fuses with parent number 2's egg over here, and now the offspring will have 4 alleles. And the way you figure out what's what is you combine the eye alleles first and then the wing alleles. So eyes have a plus and a plus. So that's plus plus. What do the wings have? Plus plus. And you do that for each one. You do eyes and then wings. Okay, so you keep them separate. Eyes are plus, plus, and wings now are A and a plus, okay? So now in here, I'm going to make sure to keep the plus symbols in front of their recessive, but you know where the A and the plus came from, okay? And you would keep doing that for every single one. You would do eyes, wings, okay? So we have plus S and plus plus. Eyes are plus S and wings are plus A. Okay, and we're going to do that for each one. I'm going to stop writing all the extra stuff and just fill them in. If you get confused at all, just send me a remind message with a picture of this slide, and I'll go over what I just did here. Okay, so each one I'm putting eyes first and then the wing alleles. Eyes first, then the wing alleles. Eyes and wings, eyes and wings, and then we do at the bottom, eyes, whoops, wrong, sorry, my pen erased that, so eyes and wings, eyes and wings, eyes and wings, and the last one, eyes and wings. Okay, like I said, if for any of them you're uncertain where any of the alleles came from, why they look the way they do, just please send me a message and remind, and I will clarify where everything came from. Okay, so those are all of our genotypes. The last thing we have to do, so you notice there are 1, 2, 3, 4 times 4, you have 16 different little squares here with all of the genotypes. And then what we do is we map out for the F2 the different phenotypes. And again, phenotypes are what you see, okay? So you can see 4 different types of flies when it comes to a dihybrid. You can either see a fly that has wild type eyes and wild type wings, okay? Or you can see a fly that has wild type eyes and mutant wings, which we'll write as APT for apterous wings, meaning it has no wings at all. Or you could have the mutant eyes. So you can have sepia eyes with wild type wings, or sepia eyes with mutant wings. Okay, and so we're going to look at our little squares on this Punnett square. We're going to look at the individual little squares to figure out which ones have wild type eyes, wild type wings first. Okay, so we have 1 wild type eyes, wild type wings. What we're doing is we're looking for any of the ones that for both traits have at least 1 plus symbol, because that's dominant. 2, 3, 4, all of these, notice each trait has at least 1 plus symbol. Okay, so that's 4 wild type. This one as well. The next one we won't circle because it's got 2 apterous alleles. Then we're back to, again, 1 plus and 1 plus there. Another one that's wild type for each trait, wild type for each trait, wild type for each trait, and then all the rest have some recessives. So then we count up the ones that we circled. So that's 1, 2, 3, 4, 5, 6, 7, 8, 9 out of our 16 little squares are fully wild type looking. Okay, and then next we're going to look for the ones that are wild type eyes and apterous wings. Okay, so we're going to look for any of the ones that have double little a's for the wings, and for the eyes, it has to have at least one plus mark. Okay, so what do you notice? Those are 3 of them that had the wild type eyes and the apterous wings. Then you look for the flip or the reverse of that, the ones that have the 2 little s symbols, so they're sepia eyes, and they have at least one plus mark. Okay, at least one plus mark, so that would be 3 of them. Okay, so we have 1, 2, 3 of them with sepia eyes and wild type wings, and that leaves us with our 1 out of 16 fully recessive mutant. Okay, so that's why when we talk about dihybrid crosses, meaning crossing 2 fully heterozygous flies or organisms for 2 traits, you will see a 9 to 3 to 3 to 1 phenotypic ratio. Okay, and again, phenotypic being what you're actually seeing for those flies. Okay, now that's how you do the Punnett square, but in the coming slides, I'm going to show you a little easier way to figure this out based on considering a dihybrid as simply being 2 monohybrids put together. Okay, again, if you have any questions or concerns, send me a message in the Remind app. Now, before we get into more of the numbers and the laws that the dihybrid abides by, I just wanted to show you what the fully recessive mutant in the dihybrid cross actually looks like. So that one lonely little corner square, that 1 out of 16 F2 flies that were fully mutant, meaning it was apterous and sepia together, they look like this little guy down here. Okay, notice that it has no wings, which is why it's apterous, and it has the dark eyes, which is why it's apterous sepia. Again, notice that it is named after its mutation. If you see a fly only named apterous or only named sepia, that means that they only have one mutation. This fly has both names as one big name, and so that tells you it has 2 mutations. So now when we talk about the dihybrid cross, you'll notice that it actually demonstrates 2 main laws of genetics and inheritance. The first one is the product law of probabilities. And that's basically telling you that when 2 independent events occur at the same time, the probability of the 2 outcomes occurring in combination is the product of individual probabilities. Now that's a long, fancy way of saying that when you're, for instance, doing a dihybrid cross and you're looking at 2 different traits, the probability of those 2 traits having a particular outcome is the same as taking the monohybrid probability of each of those traits separately and just multiplying it together. So for instance, if you think of the Mendelian ratio of 9 to 3 to 3 to 1, okay? When we were looking at crossing the 2 heterozygotes, so that was plus S plus A crossed with another plus S plus A fly, right? That first number, the 9, we saw in that big Punnett square that 9 out of 16 of those flies had wild type eyes, wild type wings, right? Well, instead of drawing out that full big Punnett square, what you could have done is look at the monohybrid probabilities, okay? So take wild type eyes as one monohybrid. What's the probability of getting a wild type fly in our monohybrid cross Well, remember that little Punnett square was 3/4 wild type, right? Remember that 3-to-1 ratio? So if you look at the monohybrid, 3/4 wild type eyes, and you multiply that by the monohybrid Punnett squares probability of getting wild type wild type wings, that was also 3/4. And what does that give you? 3 times 3 is 9. 4 times 4 is 16. Okay, so you're able to get to 9/16 by simply looking at the monohybrid ratios of each individual trait multiplied together. It works for each of the other parts of that 9 to 3 to 3 to 1. So for instance, let's take a look at how there were three of those squares out of 16 had sepia eyes and wild type wings. Well, what's the probability of getting a sepia fly in a monohybrid cross? Well, let's just remind ourselves, you can do a little Punnett square of a monohybrid cross for eyes, right? 2 heterozygous flies, gametes, crossed together. Okay, we have plus plus, plus S, plus S, SS. How many out of those 4 are sepia? 1 out of 4, right? That's why there was 3-to-1 ratio for monohybrid heterozygotes. So all you would do is 1 out of 4 sepia times, what's the probability of wild type wings? Well, we already said that wild type is 3 out of 4, okay? So all you have to do, using that same Punnett square we just did, boom, we change it to apterous, right? We change all the S's to apterous, and what do you notice? 3 of those flies are wild type. So 3 out of 4 are wild type wings. So by going monohybrid and monohybrid probabilities, you multiply them together, what do you get? 1 times 3 is 3. 4 times 4 is 16. 3/16 are sepia wild type. So you didn't have to do the big, scary dihybrid Punnett square. All you have to do is multiply the individual probabilities trait by trait. Okay? If you're having any trouble with that, please let me know in the Remind app. I'll give you some other examples or clarifications. Okay? That we just drew out is representing the product law of probabilities. Okay? And whenever there's probabilities, you will usually see some sort of form of multiplication. There's also the law of independent assortment represented by the dihybrid cross, which we've already mentioned last time. That's basically saying that when an organism is making its gametes, the gametes can separate the alleles for traits can separate however they want into the gametes. So notice when the organism here is making its gametes, the dominant alleles do not have to stay together. As long as each gamete has one allele for trait 1, one allele for trait 2, then it doesn't matter whether dominant is together, recessive is together, they can assort however they want to, okay? Which is very important because this law here, law of independent assortment, allows for what we call biodiversity. Okay, that's why you have so many variations in organisms. Okay, dominant alleles don't have to separate into only one gamete. Recessive alleles don't have to separate only into one gamete. You get variety. Okay, if you have any questions or concerns, again, just contact me in the Remind app and I'll clarify things. Now, what's interesting is that with the dihybrid cross, you can also do a test cross like we mentioned with the monohybrid. And whenever I say test cross, you should remind yourself, well, what do we do in a test cross? We cross an unknown genotype organism that looks dominant. We cross it with a fully recessive other organism in order to see, okay, what offspring do we get? And that will tell you what that unknown dominant genotype ended up being. Okay, now it's a little bit more complicated with the dihybrid cross, the various options for this, but ultimately the idea is the same, that you are trying to figure out what genotype a wild type looking fly has. And so in order to do that in this scenario, you would cross it with the fully recessive fly. So if it's dihybrid, then you're looking at eyes and wings. So the fully recessive fly would be SSAA. And that will show you, once you make that cross, you look to see what offspring you end up with, and that will tell you, was your fully wild type fly this genotype, this one, or this one? There are 4 possibilities for the genotype of a fly that looks fully wild type, which means red eyes, regular long wings. Okay, so you have a fly that's red eyes with long wings. So what are we going to do for a test cross? We're going to cross it with a sepia apteris fly and see what happens. Now we're going to draw out what we would actually look at or look for in this exact test cross. So just got to get the pen ready. So there are 4 options when you look at this test cross. You have option 1. Okay, with option 1, if that fully wild type, okay, so fully wild type, you want to know what genotype. So you're crossing it with a sepia apteris fly. So option 1 is, if that fully wild type looking fly has a genotype that's all wild type alleles, we'll write it out this way. And remember to always think of it as eyes, wings. That's why you have those 4 alleles, because 2 of them are for eyes, 2 of them are for wings. Now what you're doing is you're crossing it with the sepia apteris fly, and if that is what your wild type fly is, then what do you notice? For every trait, no matter what, this first fly is going to give a wild type allele. So for eyes and for wings, that unknown fly will give a wild type allele. The other fly for eyes will give sepia, and for wings will give apteris. Okay, so whenever you write out your 2 traits, and then write out the alleles from parent 1, parent 2. Okay, parent 1 for wings, parent 2 for wings. Okay, and that will help you keep everything organized, figure out what's coming from what. Now there's another possibility. Maybe that wild type fly is, sorry, that was supposed to be an arrow. Maybe that wild type fly is homozygous for eyes, but heterozygous for wings. Okay, and the way you'll know this is when you do the test cross, what kind of offspring will you get? Well, some of them will inherit the wild type, both wild type, from that parent. Okay, so again, we're doing eyes, wings. So some of the offspring will get wild type from parent 1, wild type eyes, wild type wings. And then from parent 2, what are they going to get? No matter what, S and an A. Okay, what else? What other flies can happen? Well, parent 1 can only give wild type for eyes, but look, it has 2 different options for wings. So what if parent 1 gives wild type for eyes, but for wings it gives Apterus? Well, no matter what, the second parent is going to give S and A, okay, for eyes and wings. So what happens in this scenario, half of the flies will look fully wild type eyes and wild type wings. Half of the flies will have mutant wings. So since the eyes are wild type here and the wings are mutant, we would call this fly Apterus. Okay, so half the flies are Apterus, half the flies are wild type with this option when it's heterozygous for wings. If instead it's heterozygous for eyes, then you'll know that because in the results you'll suddenly have half wild type but half sepia. And so we're going to draw this out now. If it's heterozygous for eyes but homozygous now for wings. Okay, so in the second option, it was homozygous for eyes, heterozygous for wings. Now we're going to look at what if it's the opposite. Again, it's a test cross, so it's always crossed with the SSAA fly. And in this result, half of the flies that result from this cross will have plus from parent 1, plus from parent 1 for wings. And then the other allele is always S, A, because that's all this guy can give, okay? And half of the flies, if they're not getting the plus for eyes, they're now getting the lowercase s from that parent, okay? So now it's giving S, and for wings, it can only give a plus, okay? So we're looking again, that's parent 1, and then parent 2 always gives S and an A. And what did I say before? That this half of the offspring will end up being sepia. Okay, so half wild type, half sepia. Then the last one is option 4. And in option 4, what if that unknown wild type fly is fully heterozygous, meaning eyes are heterozygous and wings are heterozygous? Well, what will the offspring look like now? Well, you keep doing what we've been doing. Okay, you're crossing it with that fully recessive fly. In the first set, again, we're doing eyes, wings, and what do you notice? For eyes, you can get wild type from that parent. For wings, you could get wild type from that parent. Okay. No matter what, parent 2 is always given S and an A. Or what else can you get? For eyes, you can get the plus, but then the parent 1 can give wings, being Apterus. Okay, so parent 1 can give a plus and an A gamete. And what will the second parent give? Always S and A. Okay? Or there's still more. What if instead parent 1 is giving the sepia mutant allele? Okay, so for eyes, if it's giving sepia, it could then for wings either give plus or A. So plus, again, the second parent always giving S and A, or it can give that sepia allele, okay, for eyes, but now instead it can also give the Apterus allele. And once again, this second parent is always either giving S or A, So that's what you end up as the last option. So notice for the fully heterozygous wild type, you get 4 different options, okay? You'll end up with seeing a big variety of flies. So you have 4 different options. 1/4 of the offspring will be fully wild type. 1/4 are apterous. 1/4 are sepia. And 1/4 are the full double mutant, sepia apterous. Okay? So basically, when you do a dihybrid test cross, ask yourself, do you see all the offspring are wild type when you cross that unknown wild type fly with a fully sepia? Are half of the offspring wild type and half apterous? Are half wild type and half sepia? Or do you get all 4 variety? Okay, if you're capable of getting all 4 variety, that tells you you have a fully heterozygous wild type fly. If you're only seeing one of the mutants, that tells you that for that trait you are heterozygous, but the other trait, your fly is fully homozygous wild type. So in this case, you notice only the wings are heterozygous, so you suddenly see apterous flies. If you're seeing sepia flies, well, that tells you only that eye trait is heterozygous. And then, like I said, if all 100% of the offspring are wild type, then you know that you have a true blue 100% homozygous fly wild type for both traits. Okay, so I know this is a lot of information. If you have any questions on this test cross, just let me know. I could give you some other examples, but basically be aware of what does it mean when you're doing a test cross. Why are you doing this test cross? What are you doing with it? And what are some of the options, okay? And keep in mind, like I said, this can be done for monohybrid or for dihybrid. Okay, the last thing I want to show you is just a little bit more math. It's the fact that Mendel's principles can be used to look at the inheritance of multiple traits beyond the dihybrid as well. And so basically you're going to analyze a problem like this with multiple inheritances or multiple traits by using the law of probability. Okay, now the figures and all look very scary, but we're going to do this rather simply. Okay, so I'm going to show you now how to draw out a multiple trait probability problem. So let's say you are looking at a cross of capital A, lowercase a, capital B, lowercase b, capital C, lowercase c as one parent. And it's crossed with capital A, lowercase a, so dominant recessive, dominant B, and dominant C. So you have 2 organisms. You're looking at 3 different traits. And I could ask you, what fraction of the offspring are predicted to have the genotype of capital A, capital B, lowercase b, and capital C. And the way you would do this is look at the 3 traits separately as if they're monohybrid crosses. So you can make 3 different Punnett squares, okay? So the first Punnett square would be the parent that's capital A, lowercase a, crossed with the first trait of your second parent, who's also capital A, lowercase a. Okay, it's a regular monohybrid cross, so you've seen this kind of ratio before. Okay, then you make a second Punnett square for the second trait, which in this case you have heterozygous for B crossed with homozygous dominant for B. Okay, so you'll have capital B's over here, and heterozygous over here. And then you have a third trait, so you do your third Punnett square for the heterozygous C and the homozygous dominant C. And again, it looks very similar to your second Punnett square. Now we want to know what fraction of the offspring are predicted to have this genotype here. So first we ask ourselves, what fraction are capital A, capital A? Well, that's 1/4. What about what fraction of the offspring are predicted to be heterozygous for B? That's 1/2. Okay, because it's 2 out of the 4 squares. Then we have homozygous dominant for C. And again, that's also 1/2 of the Punnett square is heterozygous for capital C. So now all you do to get this predicted genotype is you multiply your 3 probabilities. Okay, the individual probabilities, the probability of AA, 2 capitals, the probability of the heterozygous B, and the probability of the homozygous dominant C. And you get 1/16 for this genotype. I could then instead ask you probability for heterozygous A, and then the remaining traits that we had above. Okay, and you would do it in the same way, except now, what's the probability of heterozygous in our first Punnett square? 1/2 of that Punnett square is heterozygous for A. The other values stay the same. Okay, 1/2 of the second Punnett square is heterozygous for B. 1/2 of our third Punnett square is homozygous dominant. So now the probability for this genotype would be 1/8. Okay, and I'll write out that genotype as well. Okay, so as you see, you can do as many. I could have even added a 4th, a 5th, a 6th trait, and all you keep doing is looking at each of the traits individually as a monohybrid Punnett square and just multiply the probabilities together. Again, if you have any questions, any concerns, you want some more practice problems, please just send me a message in the Remind app. You could even send me a picture of the particular slide that you need a little extra help with. Okay, so I know that was a whole lot of math, so you can now take your break and then take a chance to head over to your post-lab problems and try to make sure to complete those and submit them on time. Okay, as always, if you have any questions, concerns as you're going over the material or as you're doing the post-lab assignment, feel free to ask me, because again, remember, the post-labs are not a quiz, so you are allowed to ask me for help and you are allowed to use the narration. In fact, you're encouraged to use your slides and narrations. Okay, thank you and have a wonderful day.